The homeostatic mechanisms that animals use to keep their water in line with the environment are also involved with nitrogenous wastes. Let's look at animal osmoregulation. Be able to define osmoregulation, excretion, filtration, filtrate, reabsorption, secretion, protonephridia, kidney, and countercurrent multiplier. Compare different systems of excretion, what is excreted, the organs involved, and osmoregulators versus osmoconformers. Relate osmoregulation to homeostasis and list, in order, the processes of excretion. And today we're looking at kidneys, but we're not going to be defining all the kidney anatomy. So what is osmoregulation? Well, aquatic animals may actually be the same osmolarity as their surroundings. You may remember such terms as isotonic, hypotonic, and hypertonic. Well, some organisms are simply isotonic, and they don't have to pump out water. But if an organism is not isotonic, then it will constantly either have to take in or give out water. And we see that with these contractile vacuoles in these ciliates, the paramecium. They have to constantly bail water because they're constantly taking on water because the environment is hypotonic to their cells. Terrestrial animals cannot have the same osmolarity as air because air is very dry, and terrestrial animals will need to regulate their osmolarity. Nitrogenous wastes just happen to be secreted as part of osmoregulation. Animals can be osmoregulators or they can be osmoconformers. As I mentioned, if you're isoosmotic to your environment, then you're an osmoconformer. You're just the same osmotic balance as the environment. But most organisms are osmoregulator. They're going to maintain the osmotic balance that is not the same as the environment. And we're not really going to look at plants, fungi, and bacteria here. We're going to focus on how animals osmoregulate. So what are the challenges animals face, especially terrestrial animals? Well, they may be gaining water or they may be losing water. So some organisms drink and some organisms do not. Most organisms drink, but here we're looking at a kangaroo rat, which is actually not drinking. It ingests some water in the food and the rest it can get as a byproduct of metabolism. Remember, oxygen is the ultimate electron acceptor in metabolism, and it takes 2 protons to make water. And that is going to give some metabolic water. Humans tend to drink more than kangaroo rats, and they get most of their stuff from water. They are going to lose water as well in the form of evaporation, and for humans, a lot more urine. So this also loses salts, though, because part of an osmotic challenge is gain or loss of salts. And we have to balance the two. So gaining salt means gaining more water, and losing water can mean losing salts. So how do fish do this? Well, in a marine fish, there's a gain of water and salt from their food, and there's gain of water and salt from when they drink seawater. They excrete salt ions from their gills, and they lose water from their gills and other parts of their body surface. So they are constantly losing water. The environment is hypertonic to them, and they will excrete salt ions in small amounts of water in their small amounts of urine. So marine fish are constantly drinking and constantly losing water through osmosis. Freshwater fish gain water and some ions in their food. They take up ions in their gills as opposed to excreting them from their gills, and they gain water through their gills and parts of their body surface because their environment is hypoosmotic to them, hypotonic to them. They excrete salt ions and they urinate a lot. So remember, freshwater fish don't drink. They just take up water from the environment. Marine fish do drink and lose water to the environment. And as they lose water, organisms are going to also get rid of their nitrogenous wastes. So why do we have nitrogenous wastes? Well, the metabolism of amino acids and nitrogenous bases leaves nitrogen. And that nitrogen has to be rid of some way because ammonia is toxic to organisms. Now, you can just secrete ammonia directly. And if you just excrete ammonia directly, then you just put it out in the environment, and that helps if you're living in water. But you try to keep the water from getting too much ammonia. That's only really a problem if you're living like in a fish tank or something. Most amphibians, mammals, sharks, and some bony fishes excrete as urea, which is less toxic and requires less water to get rid of. And many reptiles, insects, and land snails, also birds, are going to excrete uric acid, which is much less toxic and requires far less water to get rid of, but also requires more energy to make. So this is one way that birds conserve water, is excreting uric acid, and mammals cannot use this pathway to conserve water. They have complex kidneys. So let's take the basics of excretory systems. So you have some basic terms here. You have something called a filtrate, and that's going to be given off from the blood in a process called filtration. So you have filtrate made of water and small solutes that comes out of the blood. Now, reabsorption is when the useful molecules and water are taken back up from the filtrate to the blood. Secretion is when non-essential solutes and waste are actively transported from the blood to the filtrate. And excretion is when filtrate is going to leave the body. So let's look at different systems for this. In your basic planaria worm, you have what are called protonephridia. And these are tubules in the body where interstitial fluid, which is not blood, but just the fluid in the body, it's filtered. Flame bulbs will move the fluid. There's a minimal amount of reabsorption and secretion into the tubules, and then the water will be excreted from the body. It's mostly water. There's not a lot of solutes that are going to be lost through this. Invertebrates such as arthropods use Malpighian tubules. They take up the filtrate and secreted ions into these Malpighian tubules, which empty directly into the midgut at the end of the midgut. The colon will then reabsorb some things, especially in the rectum, that's going to reabsorb ions and water, and excretion is done alongside defecation. Interesting fact, insects don't really pee. They just kind of poop out their urine. The same is going to be true with birds and their cloaca. The mammalian system, however, is what we're going to focus on today, and that is the kidney. It's the organ where filtration, reabsorption, and secretion takes place. Excretion will occur from the urinary bladder to the urethra. There's a lot of blood flow to the kidneys, a lot of energy required in the kidneys, and this allows for changes in filtrate concentration to match the needs. You're probably familiar with the fact that your urine does not always look the same because your body needs to either get rid of less water or more water. And kidneys are very efficient. They're energy expensive, but they're very efficient. How are these built? Well, we're going to look at the cortex and the medulla. The renal cortex is the outside of the kidney, and the renal medulla is the interior of the kidney. There's also the renal pelvis, which is where the filtrate is going to exit into after it's been properly filtered. The ureter will take this filtrate from the kidney to the bladder, and, well, bladder will take it to the urethra and out. In the kidney, there are nephrons. There are cortical and juxtamedullary nephrons. The cortical nephrons are entirely in the renal cortex. The juxtamedullary nephrons go into the renal medulla. And the renal medulla is saltier than the cortex. So, since the renal medulla is saltier than the cortex, you're going to regain more water from juxtamedullary nephrons. And the ratio of juxtamedullary nephrons to cortical nephrons is determined by the organism and changes the osmolarity of the filtrate. This means that different mammals have different types of urine with different concentrations of urea and ions. And different animals are differently efficient at taking up water from their environment. So what's a nephron actually look like? Well, here's where you take the deep dive into anatomy, and you'll see none of those things are in bold because we're not going to memorize all of those. You have the Bowman's capsule, and this is where you have the glomerulus, and this is where your blood comes in, and the blood is going to lose a lot of its ions and water into the filtrate. This is where filtrate starts. The proximal tubule is where the filtrate will go through first. And you see how there are capillaries right next to that? Those capillaries are taking back up water. The loop of Henle is, well, we'll see that in a minute. It's where a lot of reabsorption occurs and also where some secretion begins. The descending limb has reabsorption, and the ascending limb is going to have some secretion. And then that's going to go past the distal tubules, which go again past arteries. And the collecting duct will collect this, and will again, you're just going to see some secretion into the collecting duct. There's a lot of vasculature here because the blood is in constant contact with the nephrons, taking back up ions and secreting ions. Here's how this looks as far as osmolarity goes. You have your Bowman's capsule with blood at about 300, how concentrated salts are in things. And the proximal tubule, you see all that reabsorption. So that reabsorption there after the filtration in Bowman's capsule is of water, potassium, nutrients, because sometimes nutrients will end up in there. The descending limb of the loop of Henle, you have reabsorption of water. And your thin segment of the ascending limb, you're actually going to reabsorb sodium chloride passively, and then you're going to reabsorb it actively in the thick segment of the ascending limb. the distal tubule is going to be more reabsorption in the distal tubule. And again, remember how the proximal and distal tubules, they both go close to one another and they're both associated with vasculature. That's so those can reabsorb. And you see how the osmolarity changes from 300 down to 1,200. And then as you lose salt, it's going to decrease again to 100 in the distal tubule. And then that's going to have the osmolarity of the interstitial fluid will go back up as you go down the collecting duct. So what happens here, it seems like it's not really getting very far, but it's taking out water and taking out sodium chloride, and all the other things the body doesn't want are now going to be concentrated in there. And this is where you can see a countercurrent exchange. The descending limb increases in osmolarity as it goes down, and the ascending limb decreases in osmolarity as it goes up. And as it's losing sodium chloride, the thick segment of the ascending limb is making the outer medulla saltier, which causes more water to come out of the descending limb. This is a countercurrent multiplier. When active transport gets sodium chloride out of the thick segment of the ascending limb, causing water to come out of the thin segment of the descending limb. So how is homeostasis maintained in all of this? Well, the body has a set point about how osmolarity should be, about 285 to 295 milliosmoles per liter. And the hypothalamus is going to be your control center here. So let's say there's a stimulus. Your blood osmolarity increases like after you sweat a whole bunch. Okay, so there's going to be sensors in the body are going to send a signal to the control center in the hypothalamus, causing the release of antidiuretic hormone, which is going to prevent you from urinating. Antidiuretic hormone is also going to make you thirsty, so you want to take a drink. Drinking water reduces blood osmolarity, and picking up more water through H2O reabsorption is going to also reduce blood osmolarity and bringing things back to their set point. So this is the response, is thirst and reuptake of water, and that's going to move things back up to normal, thus maintaining osmotic homeostasis.